Thanks for coming. And let me introduce myself. I'm Ramesh Jagadeesanalso from Recipharm. A- And today I'll share my thinking on the analytical innovation of thinking in analytical service, techniques to reduce the time to market. So the analytical, so just to give a couple of slides about Recipharm. So Recipharm is a Swedish-based company, and we have about 6,000 employees across the globe, and more than 500 products, which is more than 2,700 presentation, and we supply across to the more than 100 companies, 100 markets on the world. And closely, we have been about 650 million euro company. And we have 20 plus manufacturing plus 7 R&D facilities within the Recipharm group. So our footprint in the global arena, predominantly in Europe, and we also has facilities in US and India. And most of this, wherever you see that there is an M, it's a manufacturing, D is the development services. So we have overall 24 facilities in manufacturing, and 7 of the facilities on the development. So we offer service in 2 sectors. One is in development and manufacturing. Development from drug substance, drug product, analytical and clinical supply, and manufacturing finished product and packaging and serialization. With that introduction, I'll get into the presentation. Okay, today just to cover a few of the topics like importance of innovation during the analytical chemistry into the solution, and how to market can be reduced during the analytical testing stage, and couple of case studies which will be shared on the different stages of the analytical development, and then why knowledge on the entire product development has the value for the manufacturing, value to the analytical development. So getting into that. So innovation, so it's not that everything we have to discover, but anything is turning idea into a solution, or which makes like anything that is new and easy to assess. It can reduce our time, or the execution of the process can be considered. And then systematic approach and the execution of the process also can be an innovation during your process innovation. And the training on people, like a right training for the right people, can add a value to your time to reduce the time to market process. So with this, we can get into the things like innovation in making a quality product possible only with the forward thinking and the innovative mindset, because during the development you always get into the concern or problem how to resolve. So you need a mindset of thinking forward, as well as how we can be able to come across the innovative mindset or thinking innovatively to come out of the problem. And most importantly, then we have to closely monitoring the technological advancement. So whatever you have a problem, there is a solution into the market. Only thing you have to keep our eye open to see any of the technical advancement available in the market where we can be able to adapt to our product development or method development. When you develop a method or product, we always expect the unexpected. So whatever you do that, so you expect something with a positive result, but always there is an unexpected things. When you do the development of the method, you have a interference and co-elution of the impurities, all those things. So it is not always we expect that unexpected. How to overcome on that? Example, and on top of that, the regulatory bodies have become different. It has been drastically changed on the expectation from the pharmaceutical industry. Example, the elemental analysis have been part of release, but now it has been mandatory for all the elements like a class 1, class 2A, 2B, and class 3 elements has to be mandatory before releasing the product. You have to be analyzed and you have to release. So this is what not the case in a couple of decades before. So you used to do only arsenic test and then few of the limit tests on the on basis of the titometric or arsenic test. But now it has been mandatory that you have to do that everything, and you have to measure what is the quantity that has been present, which you have been not added. So it has been the process or in your excipients. So example like ICP, MS, OES, the NMR, and XRD. This has been techniques been used for only for the drug substance in the early stage, but now the things have been changing based on the regulatory requirement. We have been getting used this instrument for our formulation product. So we are taking as some of the external like other product development like APA techniques we have been using to the formulation, and this is being become a requirement. As well as the instruments are progressing with the techniques, as well as it is increasing sensitivity, productivity. Like UPLC is one of the reducing that a lot of time for us to reduce some analysis time. And the QDA detector will cover in the couple of slides. Then how this adds a value, because the impurities have been, which are the unknown impurities, shows more than 0.2% of the threshold in ICH guideline. Now that has been measured by using this kind of techniques. Then reducing the time to market, how? So what are the techniques we can able to use? So bringing the right process into place, right system into place, where we can able to reduce the time for the market. Add a value, reduce the time for the development, reduce the time for the validation, and bring down the speed, which can be able to reduce your time to market. Reduce regulatory burdens. So like we do the most of the studies now, ICPOS or the elemental analysis, everything. Now it's become it's solving most of your problems before going to the regulatory bodies. So you have a data, everything in before in hand, before it going to the registration, and that reduce the burden as well as the safety of the product. And enables safety. So example, as we said, once again I'm taking the example of elements analysis, which has been, all the limits have been set, any finished product goes out, then you've been testing all the elements of that, and you are confirming this is the final, the product as minimum or which is acceptable limit of the elementals in your product. So coming to a couple of example how this evolution of analytical techniques. We used to do only the titrations on a manual basis. Then slowly it has been gone into the non-aqueous mode. Then from the non-aqueous mode to we gone into the functional group testing. The functional group, after functional group, now it has become automated instruments. Now the titration, manual titration become 21st CFR compliance systems. So example, so this revolution of instrumentation and techniques, which has been adding a real value for us, the time to market. Example, we have done a couple of projects like solutions, mouthwash, gargles, and foamers. So we used to do a lot of testing onto that, which is potentiometric, iodometric titration. So we used to do only for in a day 6 to 8 samples, and we have to be there. The analyst or the scientist has to be there in front of the instrument only. Now things have been changed. So we got the automated instrument like HPLC. You can load all the samples onto that, and then you can run that sequence, and the next day you will get the complete results. So which is once again, we can sign it on electronically, and the data have been good enough to submit for any of the regulatory bodies, which has been increased at least twice the number of sample analysis. So this is a key to add the number of sample analysis so that we can able to produce or deliver the project in faster mode. And we don't know what is going to be the next. So only now the titration automated, we don't know how it is going to be the next stage. Same example of, we used to do a lot of HPLC analysis. Example, sorry. So HPLC, when we used to do the HPLC, it is in a different superficial porous particles, which is vary from the particle size of 3 to 10, And the typical runtime of 30 minutes to 90 minutes. Some of them goes more than 100 minutes based on the product requirement. And then the sensitivity was moderately low, and the detector use was large flow rate and large detection cells, and kind of resolutions were low. And in a day, mostly we used to do about 6 to 7 samples is on and average. But things are changing. So there is an UHPLC came into the place. Then how we have been adding the micron size have been reduced, the runtime has been reduced, so that we can be able to increase the number of samples as well as the resolutions are much better. Now we use most of our applications, the UPLCs, which is used as a 2 micron particle size with a different range of particle size, and then the runtime has reduced to 2 to 10 minutes, maximum of 10 minutes. And then high sensitivity as well as detector used, and which increase the number of throughput of the samples. So this way we add the value. So it is not on a great value addition for as an instrumentation, but it adds lot of value addition to analyze the number of samples. When you analyze the number of samples, you can take the more number of trials where we can be able to speed up the process and you can give. And the turnaround time for all the sample delivery, sample analysis results, it's become less than a day. So when you give the samples in the morning, it's between in the formulation analytical, you get the end of the day or maximum by next day. So in this way, it's the productivity has been increased. So it adds a value to reduce time to market. So it's a time to market is an entire process. It's in every step you keep reducing your time, then you, end of the day, you will summarize how soon you can be able to deliver the product. Then what is next? We don't know. After UPLC, other things, maybe some techniques come then which can reduce our time and which can add the value for us. Okay, and top of that, like detectors. So we were using initially for HPLC only for the UV and photodiode array detectors, like a PDA detectors. But nowadays, all the remaining non-chromophore chromophore molecules, we used to go with any other techniques like GC or ion exchange chromatography, or those kind of things. Now things have been changed. We get a lot of other detectors in the market, which adds a value to check all this in the single analytical method like HPLC. Like UV RI detector become a universal detector where most of the things where we can be able to use, and then fluorescent detector and QDA. So this is one of the value addition when you are doing for the product development. If there is an unknown impurity is been found in that your product development, which is more than 0.2%, so we can be able to check that during your development process, like a prototype stability itself, understand what is the structure of this, what is the mass of that, and where it is forming, what kind of the impurities coming from. Is it a excipient interaction or based on the stability? So which adds the value for us. So basically, I feel this is also value addition to the reduction of the time to market. And yeah, there are a couple of other detectors which has come for CAD and Corona, but based on the product we use. But now the availability of the range of detectors have been high, and which is really adding a value for us in our lab to reduce the time. And finally, most importantly, people training and training on innovation analytical techniques is the key, because there are so much of instruments, we can't able to use the one analyst to all the techniques. So we train them, the scientists, on specific to the techniques. Example, if the people have been using for ICP-MS or ICP-OS, we be trained for particularly for those, and some people been trained for the like a UPLC and the GC techniques. So this kind of specific training is a value addition to improve the productivity. And the knowledge sharing during the technology transfer as well as throughout the product life cycle. So example, when we develop the product and you develop the method and validate, and then doing the transfer is only a bit of the pieces. So sometimes the transfer takes minimum of 4 weeks to 8 weeks, because it's almost equivalent to your method development process. The reason behind, because different labs has the different process, So what we do in our end, so we share the product development summary, kind of knowledge data sheet. Along with the method transfer, we make a single couple of page data saying the how during the development how the method was behaving, and if there is any challenges on the sample preparation technique, as well as during the validation what kind of challenges it has been faced, if there is any failure, how we have been overcome, those kind of things. So when we give this during the method transfer, the method transfer lab can easily understand the history of the project, how it has been, method has been developed, how it has been validated, what kind of crisis they've been faced during the development and validation. And because sometimes we transfer to the lab with only one scientist, but over a period, the same lab will use for a couple of other scientists to execute the methods. So in that time, the knowledge sharing is becoming a bottleneck. So when we make this kind of spreadsheet, like saying for a knowledge sharing sheet, which really adds a value and during the life cycle of the project. Then upgrade the knowledge, practical tips, learning from the failure. So that's what. So when you have been executed any of the methods in your lab and during any other scientist, like in general, we follow a method development and followed by the before validation, there is a team will execute the analysis on the method which has been developed, then validation, from validation to the stability. So then stability will be transfer to the commercial. So the method is being verified, checked within the lab with the three different scientists. So it proves that robustness and ruggedness of the method. And then continuous update on the regulatory and the pharmacopoeial changes. So this is a key when you have a more number of people in your team and looking for their own project. So we have a compliance team who looks after this to make the continuous changes on the update on the regulatory and as well as pharmacopoeia, and it has been issued as a monthly internal knowledge sharing information. So which gives a lot of information for them, what kind of changes has been made in the pharmacopoeial or the regulatory requirement to get that update. And easy knowledge sharing. So since we have been, example in the Recipharm, we have been sites across the globe. So we use tools like a SharePoint. So if there is any things, whatever we share, that we come across anything new things or we come across any failure kind of information, we register that and put it in the SharePoint so that other sites of within the Recipharm team can be able to view and review and get understand from the knowledge from that. So we use the SharePoint on this thing. So example, now I'll just give a couple of examples of method development. What is our approach on getting into the method development? So we use basically the experience, because we have been using, being a CMO and CRO, so we do plenty of exposure in the different molecule and complexity of the molecules. So when we have that experience on that expertise, we apply the innovation. What is the learning from the previous project, how we can be able to adapt those kind of innovation to the current project. And then speed. So we always say that we have a different timeline, how much time we can be able to develop the methods. What is the timeline we can be able to give? So we have a predefined timeline within internally, and then we develop the methods and QBD-based approach. We use the softwares, few of the couple of softwares to do all the QBD-based approach, and we execute that. So this is and sometime when we draw the QBD guidelines, and then it has a value which meets most of the regulatory requirements, and it's good for, and most of this, the speed is a tailor-made solution. So it's not for the it's not for the common for everything. We have a different approach for like a dissolution and assay and related substance. We have a certain procedure how to execute that in the tailor-made option. Just given to give a small case study about the development, what we do. So since we don't have our own product, so we get the requirement or response queries from the customer. We have, this is the new formulation. So example of this product is in pretty old, about more than 15 years in the market as an OTC product. And they were using a method with three different, like one assay method, one preservative, and one RS method. So all these 3 methods, they have been running individual systems because the methods have been developed in 15 years before. Now, based on the market requirement, they have been doing the reformulation. When they did the reformulation, they found there was some interference on this existing method. When they came back, so these are the process what we can able to do. And then what we have been done is we developed a single method for all this, like one assay and preservative and degradation product all together in a single method by HPLC, which is 80 minutes runtime. If you see, what is the value addition? Instead of 3 HPLCs and you use only 1 HPLC, and then you move on, and all peaks have been well resolved, and then it's the turnaround time for that. It's in a commercial product which is available in the market. So the turnaround time for them is pretty much has been reduced, as well as the cost to the companies has been reduced. So this is what this way we can use the efficiently your resource. So this is what we had a value to the clients, and that's what we are here. So example of typical chromatogram for this, what we have been done for this, the 80 minutes, all the peaks. This we have spiked all the impurities, all the actives and preservatives. This is a typical example. Okay, the question, so is it a 80 minutes runtime is good? What's next? We came back with a method in UPLC which is on 25 minutes. So in summary, they have 3 different methods which is running 25 and 50 minutes. HPLC method is developed in 80 minutes. Now we come with the UPLC with 25 minutes. Now we are checking with because that manufacturing site does not have the UPLC, so we kept on hold, but now they are running on the HPLC, but we are ready with the method with the UPLC, which is 25 minutes. So this is the way where we add the value to the customers for their prices and their requests. Sorry. So coming back to on method validation. So method validation, so if we have both options, what we have our approach into the method validations is, once again, all the method validations have been, we take the responsibility to make the protocol and reports preparation with as per the ICH guideline, and some are specific based on the customer requirement we can make it, and this methods will be on a stability indicating. We follow strict GMP compliance, and as well as what is the thing, if we do the method validation from our end, and we also support for the quality, the queries response from the regulatory filing. If the methods have been validated from our end, it has been filed, then we do that. And it's the faster turnaround time. So we have all predefined timeline for each of the parameters, and which will be executed. We have been succeeded more than 95% of the time. The timelines have been very strict, and we have been delivered. What is the specialty here? We use everything on a paperless lab. We don't take any of the printout, everything in electronic signature. Then there is anybody is required, then we can give the soft copy, electronic copy directly to them. And we use the Empower custom field calculation, so which avoids all the data data competition, and then we can easy to share with any of the regulatory bodies. We also does lot of stability studies, and like big pharma companies have been efficiently using us, and the facility can accommodate a wider range and volume of the product. A stability sample, we have internal things within 15 working days. We analyze and we deliver with the report after the review. Once again, the speed is process is value addition for the custom fill. So every product has been done for the protocol and approval for all the custom fields, execution of that, and the electronic signatures have been done. We also had a range of stability studies, and separate few other studies like a freeze and temperature cycling studies have been done. And the knowledge on the entire product development, what is the requirement for the product development for the knowledge? So this is what makes a difference when you have only standalone analytical, and the product, if you develop for the overall, how it adds a value. So we understand the process of the product development, because the result within our team, we also support for the product development, and we also takes care of the standalone analytical. So in that way, so from the API, RLD characterization, compatibility studies, prototype development, final prototype and scale up, exhibit batch analysis and stability, then finally going to the tech transfer. So that's all. What we get into that more detail is, like, we check from the initial onwards the form by the HRD and then PSD, as well as the bulk and tab density, to understand how your raw material is, or the API is having a properties on that, and then try to get that more and more on the degradation pathway by asking that the API, the open part of DMF, to understand how the API behaves and how this works on that. Then compatibility studies, we also check that the selection of the excipients, and then selection of the container closer if there is a packing material, and reverse engineering based on the product. So we do also the reverse engineering, how the product works, and what are the excipients. If it is in a semi-solid, we follow the guideline of ICSE Q1 and Q2 process. So based on that, we can derive what is all the excipient and grade it has been used. And also during the prototype development, we do the method development for assay dissolution related substance, as well as for semi-solids, topicals, we do IVRT and IVPT studies. And then final prototype and scale up, we get into that all the mitigations of the scale up process, how this has been works during the what are the challenges it comes, and the change in the manufacturing process, because from the lower scale to the higher scale. So we get into that, and during the successful method scale up, we do that method validations till that so we observe that how the product behaves on that. And finally, like a exhibit batch, the regulatory requirements, we run that with a complete under the GMP environment, and we do the complete stability studies. And then when you submit to the regulatory bodies, if there is any question or queries, we're happy to work on that. Then formulation transfer and commercial facilities, this is all the key that we, once all the methods have been developed and validated, we transfer those to them. And then finally, just to give during the SNR process, we also come across some kind of concerns, like the product is developed and validated, still we have a concern how it is. So one of the product, the lozenges, so when we did that all the method development and validated, in the real-time stability studies, we found there is a few unknown impurity has been found. So which is not been the case, it is not come any time on the forced degradation study, but in the real stability data, we found there is in a third month, we found there are a couple of impurities. And then we are trying to investigate. By the time 6 month sample has come, then once again we found another third impurity, unknown impurities, which is more than 0.2% of the reporting threshold. So how do we come approach on this? So example, these are the typical chromatogram. If you see the first one, it's in a control sample, and second one, third month stability sample, we found there is an impurity 1 and impurity 2 is the unknown. And then 6 month we also found there is a third impurities, which has never been the case. So some of this particular few molecule very rare, and with a different formulation like a lozenges, we found this kind of issue. Then what we did, so we did the identification and characterization of that impurity, and then we identified what are those impurities. So which is we have our own, once again, we have our different sites has that capability who can able to analyze and then see that. So we did all those things. Then we did the impurity isolation and characterization, and then method validation, and we did the identification of those impurities. So these are the structures which has been found with the help of other LCMS and the NMR data. Not only this data, we have been generating more data to identify and confirm the impurities. And then once again, these impurities have been injected the existing method, and we found the peaks have been well separated, example of this. So then we did the revalidation for those impurities. So with that, just to give some conclusion on the system compliance, so we all follow the strict GMP compliance, like we have dedicated people for the projects, and as well as quality agreements in place to ensure that we have good terms and conditions to the clients and ensure. And we save that lot of time with the systems like using the Empower 3 for the custom field calculation, and paperless lab, which adds a value to minimize all the repeated work or some kind of transcriptional work which use that. And then we also use the e-limbs. So these are the value addition for us to improve our process as well as time to reduce the market. And in summary, so innovation in analytical chemistry combines with the knowledge of product development cycle as to overcome the complex challenges. So it's not only the analytical, we also get into the product to understand what is the product, how it behaves on different stages. And then technical advancement can improve the time to market, like example of the moving into the, I'm just quoted a couple of examples like from automated titration to the UPLC, but there are other techniques which is available in the market. We use that to speed up the process. And finally, define a systematic process, enable the efficient and time to delivery of the market. And then training with a strong quality and system ensure the compliance and safety of the product. Okay. So with that, I'll conclude. Thanks. So we have a separate wings to support only an analytical as an Recipharm analytical services with you guys. Thank you for you. Thanks for your time.